Now I feel like crap. Okay, so this layer where the cells, some of them start to look pointy, they call them prickle cells sometimes. In the stratum spinosum, in addition, we mentioned these before, you can find some of these epidermal dendritic cells, which are called Langerhans cells. And the Langerhans cells are immune cells. They are the most superficial of your immune system cells. So they help pathogens, keep pathogens from getting into deeper tissues. So the last of the most common types of skin cancers is found in this layer. As usual, your textbook only represents it in the later stages, which isn't particularly helpful. So let me show you what it looks like in the early stages. This is called squamous cell carcinoma. And it's a little confusing because it's called squamous cell carcinoma because they're not actually flat yet, but I had to get used to it too. This is what it looks like in the early stages. To me, it always looks like a wound that you took the scab off too soon and it's kind of irritated. And sometimes it's dome-shaped and sometimes it's depression. It tends to get dome-shaped in the later stages. But it looks like a little irritated wound that won't heal, red spot that won't heal. And then in the later stages, it gets to look like that, which looks like a freaking booger on your skin. So that's definitely not good. So squamous cell carcinoma is intermediate in severity. Well, I mean, any cancer can be severe enough to kill you, but intermediate in the likelihood of metastasis between basal cell carcinoma, which is pretty slow growing and unlikely to metastasize comparatively speaking, and melanoma, which is really fast to metastasize and pretty aggressive. So this one's kind of intermediate. Now I'm just going to reiterate that I am not saying that basal cell carcinoma can't kill you. I'm just looking at statistical likelihood. One of them is really an aggressive, fast-growing cancer. Go ahead.
Why is more intermediate when basal melanoma are both in that?
I don't know. This one just does not metastasize as quickly, and I don't know why that is. It could be because it's more superficial and doesn't actually move into the deeper tissues. But I don't know. It's a good question. I don't know if we know the answer to that. At least I don't know the answer to it. If I remember to look it up, I'll teach it to you, though. Okay, so that's squamous cell carcinoma. And then the last two layers are these two up here. And so I have the stratum granulosum, and notice it starts to look grainy, and I'll show you actual histology slides in just a second so you can see that. Oh, last three layers, sorry. So the stratum granulosum. So let me show you the histology slides so that you can see. It's easiest to see, everything's easiest to see in thick skin because you've got a lot of space to look at. So let's move up. The stratum basale is always pretty easy to find because the basement membrane's right underneath it and the tissue totally changes type. And then everything from there to where you see this dark pigmentation, that is all stratum spinosum, right? And then this starts to get pigmented. It's not melanin. What it is the precursor to hyaline is called keratohyalin, and it stains with these staining techniques. So it begins to look granular. What you really have in these cells is a whole bunch of little vacuoles of keratohyalin, which tend to stain darkly. So this is called the stratum granulosum. And the cells are kind of sort of specializing in production of that protein. Now you can see this a little bit on the thin skin, but it's a little harder. So let's look at the thin skin. The bottom layer is still pretty easy to find, right? That's the stratum basale. And then between there and when it gets dark up here, that's the stratum spinosum, right? And then right here before you get to the dead cells, that's going to be the stratum granulosum. But I don't usually make you label it in thin skin. The bottom and the top layer are fair game in thin skin. The rest are a little hard to distinguish from one another. Okay, so that's the stratum granulosum, and then we are going to move up to the stratum lucidum. And lucid means clear. So what you have here is I've already made all of the keratin, and then all of the organelles of the cell die off. And now I've got pretty much a dead cell membrane bag that's filled with keratin. And right before they become kind of crunchy, they are in the stratum lucidum. And so with the stratum lucidum, you see lighter cells, or sometimes clear cells. And so you can see that right here, right? Basale, spinosum, granulosum, and do you see the lucidum right before you start having the dead cells? So that's the stratum lucidum. And then the rest of this is going to be the stratum corneum, and that's dead cells that are filled with keratin. You can't really label the stratum lucidum on thin skin. Now, conceptually, it has to be there, right? Because the cells had to die off at some point. But you're never going to be asked to label it there, right? So you do produce the precursor and then the cells die off, but you're never going to be asked to label it there. Okay, so the last layer, the top layer, is the stratum corneum. And the stratum corneum is this layer of dead skin cells filled with keratin. It's a waterproofing layer. And corn, like unicorn, horn, this stuff is made of primarily keratinized cells. It's called a waterproofing protein, but really it's a water-resistant protein. And it is on the surface of your skin. It's also the primary component of your hair and your fingernails. That's primarily keratin. And then in other organisms, it is what makes up scales and feathers and claws and even horns, although not antlers, like the horn of a rhino. Is made of keratin. So if it builds up, it can become really substantial and thick. On you, it's not that way. So the stratum corneum varies pretty dramatically in thickness. The stratum corneum here would be maybe the soles of the feet, right? And here would be right around the eyes. But you can still see it in thin and thick skin, right? And it is really what is the most variable in the epidermis, is the thickness of the stratum corneum. The other layers do vary a little bit, but not dramatically. Okay. It usually, in thick skin, I say right here it makes up 3/4 of the epidermal thickness, but I just mean that in thick skin. It doesn't necessarily in thin skin. Now remember with the stratum corneum, well, remember that when the cells were made, they were stuck together with desmosomes, right? And then as they move up, some of the desmosomes break, but all of them don't. And remember, by the time you get off to the top and you're exfoliating, they don't come off all the time as individual cells. They come off as clumps because they're still stuck together with desmosome. Go ahead.
Does friction determine the thickness? Like with calluses, or like if you have...
It influences the thickness, although even babies who have not had friction, they're not walking, for instance, the skin is thicker on their feet than it is on their face. But what you do with it can actually increase the keratinization rate. And so you get protective, like playing guitar, right? If you start playing guitar, you are going to get really thick calluses on your finger. It's a protective mechanism. It's kind of like the friction equivalent of producing more melanin when you're exposed to UV radiation. You're going to produce more keratin when you're exposed to abrasion. Does that make sense? But you didn't produce zero melanin to begin with, right? Or zero keratin to begin with. So, okay. So now let's talk about the dermis. We don't go into the dermis a whole bunch of detail, but there's some interesting things about the dermis. So the dermis, even in thick skin, is thicker than the epidermis. Do you see that? Okay. Do you see that the dermis is thicker? And the dermis is going to go from the basement membrane all the way to where you start having loose areolar connective tissue and adipose. That's where the dermis goes. That's got a bunch of stuff in it, erector pili muscles and sebaceous and sudoriferous glands and a whole bunch of nerve endings. As a matter of fact, most of your sensation that you feel here is in the dermis. Because remember, in the epidermis, you only had one type of sensory cell right there. And that's a tactile cell or Merkel cell, and it's pressure sensitive, which means that almost all of the sensation is actually not happening in your epidermis, it's happening in your dermis. Like, for instance, temperature. You can't tell temperature with your epidermis at all. You can only tell it with your dermis, right? You can tell a little bit of pressure. There's no pain receptors in your epidermis. So most of those nerve endings are down here in the dermis. That's where you do most of your sensation. Okay, you also have, not pictured here, but we'll talk about later this semester, lymphatic vessels there. Do you guys know anything about the lymphatic system? Most people don't. For some reason, they don't really teach it in anatomy classes before college. It drains excess fluid from tissues and eventually dumps them back into your cardiovascular system. It's also part of the immune system. So you got lymphatic vessels in there as well. And then, a couple of different things. Because it's so vascular, it can nourish the epidermis by diffusion. Because even the thickest epidermis the living part is still microscopically thin, right? Does that make sense to you? So it can nourish the epidermis by diffusion. Now here, this thin skin, this is kind of a beautiful photo because you can see what tissue the vast majority of the dermis is made up of. What does that look like? Dense irregular. So the vast majority of the dermis is made up of dense irregular connective tissue. And it's really vascular and it will nourish the epidermis from below. Does that make sense? Okay. So we got our erector pili muscles, our hair follicles and glands. We already did that. Okay, so interestingly, in certain places, well, in all places, the surface of the dermis and the epidermis where they meet, it's not smooth, right? It's actually curved. Does that make sense? But in some places, it's so very curved that the epidermis follows the dermis. You guys with me? And when that happens, it ends up making these dermal ridges, and the epidermis follows the dermal ridges, and it manifests as fingerprints and footprints when the curves are so dramatic that you actually follow. Now, I want to talk about the evolutionary advantage to fingerprints and footprints, or think about it, what were they good for? What are fingerprints and footprints good for? Yeah, exactly. Having fingerprints versus not having fingerprints, or having footprints versus not having footprints, is the difference between wearing your church shoes and your tennis shoes. Which one are you going to fall on your butt on? Church shoes, exactly. That's reason number 25 that you didn't want to go in the first place, right? It's the damn shoes. I don't want to wear those shoes. So, so fingerprints and footprints will increase grip. And if you combine that with the secretions, you got some good grip that you have there. And so interestingly, just a little tidbit, fingerprints and footprints. When I worked in forensics when I was in grad school and just after grad school, we did forensic DNA analysis, and a lot of them were paternity cases, but we also had some criminal cases. And interestingly, if you have DNA evidence in a criminal case, you can't tell two identical twins apart. So you've always got an alibi, just so you know if you're an identical twin. So if you had, for instance, a rape kit and a suspect, so you cannot tell the DNA apart from two identical twins. But if, however, you have fingerprints or footprints, they have different fingerprints and footprints. They're very, very similar to one another, basic same genetic makeup. But interestingly, they figured out that the pressure and times that you touch the wall of the amniotic sac during development impacts the development of your fingerprints and footprints. So identical twins have very similar fingerprints and footprints, but they're slightly different, and they can be used for identification. So we have any identical twins in here? Your husband's an identical twin? You're one? You're an identical twin? I'm always fascinated with identical twins. If you want to, I want to see your fingerprints and footprints. Just fingerprints, actually. I just want to see them. Have you ever compared yours to hers? Has he ever compared their fingerprints? They did? What was the study about? Were they raised separately?
No, but they're one of the subjects.
Oh, they're one of like the control group? Yeah, so I have like collected, because I'm a weirdo, fingerprints from identical twins, and I keep confirming that they're like super similar to one another, but you can always tell the difference, at least when you get enough fingerprints or footprints. It's pretty cool. So you can go compare to your sister, right? And let her know that she will be your alibi if you murder anyone. Right? She's also your organ donor, so that's really nice. Be nice to each other, right? So, okay. If you ever need an organ, if they need an organ, her organs aren't going to do you any good. Okay, so let's talk about tattoos for a second. So if a tattoo theoretically did not go past the epidermis, how long would you get to keep it? 30 days, exactly. That would be a really expensive waste of money. So a tattoo, by definition, has to go into the epidermis, unless it's, of course, like a henna tattoo, which isn't really a tattoo. It's just like dye, right, going into your skin. And henna tattoos last, I mean, they say they can last up to a month, but they usually last like a week, right? Sometimes a little longer than a week. So those are just basically dye that actually sinks into your epidermis. But an actual real tattoo in which you inject the dye actually, so you know what a tattoo looks like, what it's going to have to do is it's going to have to inject the dye into the dermis. It's going to have to be a brilliant enough dye that you're going to have to be able to see it through the microscopic layer that's left on the top, which isn't very hard. So what happens here is interesting. You have to use dye that does not cause a massive immune system reaction. And of course, there's been a lot of experimentation that's gone on with this. You can't just inject anything into your skin and not die from it. But we figured out some dyes that actually work, and you inject a dye. And then interestingly, what your immune system does is not a massive immune system response to it, but what it does is tries to sequester the dye. And it sort of tries to sequester the dye almost as if it's building a scar tissue capsule around it. And the interesting thing about it, it's not exactly scar tissue, is it keeps it in place, right? So it keeps it in place so it actually doesn't migrate. Different dyes work well, less well. There's been a lot of advances in tattoo dyes. The colors are more stable now than they used to be. They last for a lot longer than they used to. But what they are doing is basically just sort of putting the dye deep enough into the dermis that it's not going to go any deeper. It's not going to become superficial. And you have to get the dye so that it's actually visible through the epidermis, which isn't very hard because you really, I mean, you don't tattoo the hands and the feet, right? I mean, you could, but ow, that sounds horrible. But a lot of times the skin that you tattoo is not the thickest skin that you have. It's also not the thinnest. So that's how a tattoo kind of works. Now, there's several different ways of doing tattoo removal. The only one that I understand well enough to talk about is the laser tattoo removal, in which they blast you with the laser. And what the laser does is specifically breaks up the dye, okay? And then the dye will diffuse out through the tissue. But in the process, a lot of times it also damages the superficial tissue. So you can get scar tissue instead of the tattoo. So it's not as if when you, for instance, get tattoo removal, you end up with like virgin skin again. It's unlikely that you would end up with virgin skin again. A lot of times there's tissue damage as you're going through it. So you can tell that something was there. Okay, so last little creepy thing to talk about is that the dermis right here is what generally in other organisms we will moisturize and stretch and keep nice and supple and moisturize and stretch through the process called tanning. And it is that dense irregular connective tissue that is nice and strong but also very flexible that forms leather. That's what we make leather out of, is the dermis of generally other organisms, although we certainly have made it out of humans before, not me personally, okay? Not done it. People have done it to each other, both creepy Silence of the Lambs kind of situations, and also, do you guys know, I encountered this when I was in Europe the last time, so people do some creepy stuff with like the parts of the saints, so the parts of the saints in the Catholic Church, like all the pieces, if somebody was canonized and they became saint, all of the pieces of the person are holy. And so you got like some Saint whoever's little finger bone, in, a glass case with a whole bunch of jewels around it when you go visit this church, that church, whatever. But in addition, at some point they figured out that there's no reason to waste the skin, so they would take the skin of the saints and use them for the covers of Bibles.
Oh no.
For real, for honest. I've totally seen them. I don't have one, but I've totally seen them. Yes, that was super duper holy Bible, like extra holy Bible, right? Yeah, so leather, a leather book cover. It's really gross. People are weird, man. Okay, so now let's talk about the hypodermis. And the hypodermis is here, also called the subcutaneous layer or the superficial fascia. And the hypodermis is made of greater and lesser amounts of areolar connective tissue and adipose tissue, depending on how heavy you are and depending on where you look in your body, because adipose tissue deposition is not consistent over the parts of the body. So we'll talk about that in just a second. So the hypodermis is actually not part of the skin. So the skin is only two layers, epidermis and dermis, right? but it's always taught with the skin because, of course, it's relevant to the skin, but it's actually not part of the skin. It attaches the skin to the underlying organs, and about 50% of your body fat is stored there on an average-sized person. Obviously, if you're morbidly obese, it's a lot more than 50%, but we're just talking about an average-sized person. And it, of course, is going to increase in thickness dramatically in the presence of weight gain, and then you lose it when you lose weight. But it's really different how you deposit body fat in the presence of estrogen and testosterone. So let's talk about this and talk about body fat deposition changes as you go through puberty. So if you have two 7-year-old kids, right, and you're just looking at them from afar, a silhouette you really can't tell a male from a female, right? They're just like a kid, right? Kind of round in the middle and skinny little legs, you know. So the kids. But once you hit puberty, then one of the many things that changes is your body fat deposition patterns start to change. And those body fat deposition patterns start to change, and they change slowly at first, and the males hit puberty a little later than females do. But let's now fast forward to two 18-year-olds, right? And we're looking at the silhouettes of two 18-year-olds, average body size, not like excessively thin or thick. And you can tell the difference between them, right? Even from afar, you can tell the gender differences. And the gender differences are primarily due to body fat deposition changes. Of course, you have muscle changes as well, but you don't even have to be super muscular to tell the difference between a male and female. So let's assume we've got male and female, 18 years old, about the same height, right? Neither one of them is outrageously muscular. So what happens is in the presence of testosterone, your body fat deposition starts to change from, it changes to very little on the legs, and when you start depositing body fat, you deposit up here. So if a male gains body fat, it's going to be here in the chest, right? So they can get little man boobs before they stretch out, right? But when in high levels of estrogen, how girls start to gain weight is very different from how males start to gain weight. They tend to deposit less body fat around the middle, so their waists are relatively small, and more body fat in the legs and the hips, and then body fat in the breasts. So their shape is like this, right? And so you can tell from afar, it's primarily body fat deposition. And fascinatingly, there's been some cultural anthropology studies that show that men, specifically, let's talk about heterosexual men. Heterosexual men react and find attractive a certain waist-to-hip ratio that is pretty consistent across cultures. Even if in some cultures they prefer women slightly heavier, and in some cultures they prefer women slightly thinner, there is an ideal body fat deposition that you can manipulate photos, and males will all of a sudden think, oh my gosh, she's fascinating. I totally want to get to know her. It is 0.7, the famous waist-to-hip ratio that men tend to find universally amazingly attractive, regardless of whether it's a heavy 0.7 or a thin 0.7, that waist-to-hip ratio. And you can like fiddle with pictures, and they will all of a sudden find it fascinating. Women do not seem to be dramatically affected by the same thing. It's not body fat deposition with women, although you can manipulate a male's face to make it look like he has more testosterone, and you will find him far more attractive. And it's primarily bone structure in the face. Testosterone tends to make more dramatic bone structure. Although she might not want to marry him, she definitely wants to have sex with him. Right? You don't always want to choose the one with the highest testosterone to hang out with, right? Like, I've done that. Okay. And then, fascinatingly, when a woman hits menopause, your estrogen levels diminish, go really, really low. The body fat deposition will start to change again. What starts to happen to a woman unless she is battling her hardest against it with body fat deposition after menopause? Yeah, well, it increases overall just because your metabolism is slowing down, but you start to deposit body fat in the middle, and you start to lose this shape. Fascinatingly, that is evolutionarily a sound strategy, because this shape is communicating sexual viability. And this shape says, I'm done, right? It does. I mean, evolutionarily, we want to battle against it because now we just want to have sex for kicks and giggles. But, I mean, historically speaking, you have to have some way of communicating, even from afar, who is worth, you risking life and limb, right, to make some babies. And you need to know, and this is sort of telling you that. So it's interesting. So males lose their testosterone too, but so gradually that it's almost imperceptible. They don't even really start having dramatic drops in testosterone that manifest as low sex drive even until their 60s. And it doesn't even happen with all men in their 60s. And they can still father children into their 80s and 90s potentially. So, okay. So you can label all that stuff on there. And then the last two things for us to talk about, last three things. We're going to talk about what happens when you damage your skin, and then we'll talk about skin aging and burns. Okay, so this next topic is what happens when you damage your skin. How does it heal? This also is relevant to other tissues as well. We're just going to talk about it related to skin because you have more experience with seeing what happens when you damage your skin. But for instance, you can have internal damage that would have these same healing patterns as well. So integument repair and regeneration. So what's going to happen here is after you damage your skin, there's going to be this process by which you are going to repair the damage. And the ideal damage repair would be if I lost a cell, I replaced it with a cell of that type for every cell, right? If I lost the matrix, I replaced it with matrix of that type. And then if that happened, then you wouldn't even be able to tell that there was any damage, right? And that process is called regeneration, and that's definitely plan A. But regeneration is slow. So sometimes it's more important to fill up the hole than it is to fill it up properly, right? So if you have a large wound, for instance, I want you guys to think about this. If you have a large cut or wound on your arm, the size of the scar, which is not regenerative, it's changed tissue type, tends to be smaller than the size of the wound, right? Regeneration happens as quickly as it can at the edges, and then your body goes, we have a hole here, people. Put something in that hole, and it spackles the middle with scar tissue, which is called fibrosis, which is faster, but you lose functionality during it. Does that make sense? So plan A is regeneration. A, sometimes it's not fast enough. And then B, some tissues can't regenerate. Some tissues are amitotic, like cardiac muscle and nervous tissue. They can't regenerate. So you're never going to have regeneration if a tissue is amitotic. If a tissue is mitotic, you might have regeneration if the wound is not large. Okay, so let's talk about that. So with regeneration, dead or damaged cells are replaced by cells of the same type, and normal functions restored. But with fibrosis, dead or damaged parenchyma, this word, don't let it confuse you. Parenchyma are the unique cells in a tissue. So for instance, for the liver, they would be hepatocytes. For the brain, they would be neurons. For the pancreas, they would be pancreatic cells. So all organs have connective tissue and then some unique cells, right? So if you do fibrosis, which is scar tissue formation, you replace the parenchyma, the uniquely functioning cells, with connective tissue. And then you lose functionality, at least in that location, right? Okay, so it's going to fill up the hole because we don't leave holes in things. We also do not leave dead cells in things because they're pathogens that like dead cells. It's going to fill up the hole, but you're going to compromise functionality. So I want you to think about scars on the surface of your skin where you can experience them. Do they tan the same way as the tissue around them? No. Do they produce oil or sweat the same way? No. And their pigment is usually different. And do they always grow hair? No, they don't usually grow hair, right? So if you have a scar in your eyebrow, you're usually going to have the bald spot. Every once in a while, you can have them like not lose all the functionality and they'll still have some of it, but they're distinctly different than the tissue around them. And so let's look at how that process occurs. The process of fibrosis starts with increasing blood flow to the area, which sounds counterintuitive because you don't want to bleed, but what you're doing is two things when you bring more blood flow to the area. You're bringing white blood cells to whoop up on pathogens. You're also bringing in platelets, which will eventually result in a blood clot. Does that make sense? So you're going to do that, and then what happens also is you have more capillaries sprout out of the local blood vessels. That's called angiogenesis. And they're going to bring more nutrients for the healing process and more oxygen for the healing process. And do you remember those little connective tissue stem cells that hang out around blood vessels? Mesenchymal cells? Remember? Those can undergo differentiation and make some fibroblasts. And then the fibroblasts will make fibers, and they're going to start to heal the wound. If it's on the surface, they're going to heal it from deep to superficial. What kind of fibers would you use to start this healing process? Collagen, elastic, or reticular? Collagen. Starts to make collagen fibers to sort of knit the wound from the bottom to the top. Bundles of collagen fibers. It's not instantaneous, but it's going to start making them. And so in the later stages, what you're going to have is a whole bunch of collagen fibers and a whole bunch of blood flow in this area. And at some point, sometimes the scab may fall off at that point, and you have that pink fragile tissue underneath. That tissue is called granulation tissue, and it's the tissue in which you've filled the hole, but you've still got a good level of blood flow, and you're not really finished with the collagen production yet. So that pink fragile tissue is called granulation tissue. And then in the next stages, what happens is the blood vessels atrophy because you got enough, right? You don't need to be super active anymore. And then you get this pale, almost avascular scar. The scar has very little blood vessels in it, so it tends to be paler than the tissue around it. But notice that I have lost almost all functionality in the scar area. I don't have any hair, I don't have any oil, I don't have any other stuff in it. I usually have also a different texture. It's less likely to be as elastic as the tissue around it because it's mostly collagen fibers. It doesn't have zero elastic fibers usually, but it has very few. And then in the last stage, they're not showing here, what happens is the edges. When it's really finished with the healing, the edges will pull together. Have you ever felt that tightening of a scar at the very end of the healing process? It pulls together. That tightening is fine, although it can be uncomfortable. It can be problematic if it happens at a joint, right? If that tightening happens at a joint, it can actually reduce mobility. And we'll talk about that again when we get to burn victims and how fibrosis can be problematic with healing and burn victims. Go ahead.
Couple questions. So
Okay.
I have big burn scars on my wrist right here,
Mm-hmm.
and they do pull in at the edges. But when they were first like brand new skin, I would get blood. Like if I bumped it, it would be like big blood hits for like instantaneously. And is that when it was still
In the granulation tissue, it's still super duper vascular and doesn't have enough collagen to protect itself. But once you get past the granulation tissue, it does not tend to be easy to injure compared to other tissues because it's not very vascular, right? So it's not going to bleed as much. So yeah, you were just still in the early stages. And it takes a while for burns to heal because when we talk about thickness burns, you will see why it takes a while. Okay, so now, so that's how the scar works. Let me briefly talk about what happens if you scar internal organs. And the reason this is worth talking about is because, so you remember your fallopian tubes or your oviducts, right? So your fallopian tubes or your oviducts, if you have scar tissue formation occur in the fallopian tubes or oviducts, that last little part, when you pull the edges together, can kink the tube. Does that make sense to you? And that tube is relatively small to begin with. So if, for instance, you have scar tissue form on one side of the wall, right, and then it pulls, it can actually kink the tube. Now, that is one of the leading causes of infertility in women, is scar tissue formation in the oviducts. And if that occurs in a single oviduct, they're hardly big enough for a fertilized egg anyway, and all you have to do is kink it a little bit, and then you can't actually get it to the uterus. It can reduce your fertility by about 50%. And the reason that's worth mentioning is because one of the major reasons, in addition to just protecting your health right now, one of the major reasons to use protection, specifically condoms, during your sexually active years is because you are protecting your future fertility. Because a lot of these STDs that can cause pelvic inflammatory disease can cause this fallopian tube scarring, but they're asymptomatic, and you will never have known that it happened until you actually start trying to have a baby. And this is one of the major causes of infertility in women, is that exposure. So it's one of the reasons to mention it. Okay, so if it occurred on the surface, the epithelial tissue will sometimes grow over it, and so you won't necessarily lose all of the pigmentation, for instance. There may be still a little bit of melanocyte production because the epithelial tissue will grow over the connective tissue. Okay, so what are the other reasons that you could do this besides it's too big to heal quickly? Well, it depends on the types of cells that are present there. If you damage a tissue that is non-regenerative, fibrosis is your only possibility. So for instance, if you damage neurons or cardiac muscle cells, neurons are amitotic. Cardiac muscle cells are amitotic. So what happens in your heart, for instance, if you have a heart attack, is the cardiac muscle cells are cut off from oxygen and then the cardiac muscle cells die. You bring in white blood cells and they scoop up the dead cells, but we don't leave a hole, right? We do not leave an absence of cells, and we can't go through cell division. So you actually end up making scar tissue in the wall of the heart. And the important thing about that is that that reduces your contraction capacity, even if minorly, in the heart, and makes it more likely for you to have a second heart attack, right? So one predisposes you for another one because you've damaged the actual wall. The same thing in the brain, although you don't really form scar tissue with the same kinds of cells in the brain. But if I have a stroke or brain damage, what happens is those cells die off and the neurons cannot be replaced. The glial cells can, but the neurons cannot be replaced. And so your immune system and the cleanup system of the brain scoops out the dead cells, and then you can't make any more neurons. So you just fill it with the brain's equivalent of scar tissue, which is these astrocyte cells. So if you have what's called permanent cells, non-regenerating, amitotic cells, fibrosis is your only possibility. The other extreme is, what if I have a tissue that is damaged that has a really fast rate of cell division anyway? Then you're way more likely to do regeneration. And so, for instance, all of your epithelial tissues are more likely to do regeneration than other tissues because they're already dividing really quickly. Or your bone marrow, that divides really quickly, and so I can give bone marrow and then I'll just regenerate more bone marrow instead of like making scar tissue in there. So those cells are called labile cells. Those are the ones that divide all the time anyway. And then the ones that are intermediate are ones that are not dividing every day all the time, but remember how to. And these are like a lot of your gland cells and your liver and your pancreas. So if you damage those, they can regenerate. Your liver is fascinating because they figured out, I don't know, 10 or 15 years ago that if somebody needs a liver transplant, I don't have to wait for someone to die. So, I mean, they still do donations from people who have died for liver transplants, but you can also do a living liver transplant. So if my little boy, for instance, needed a liver, I could give him a lobe of my liver, and then give me about 2 years, and I will regenerate the entire size of that lobe. And give him about 4 years, assuming that he didn't reject it, and then he will fill the entire space. It'll be weird-looking liver, but he'll fill the entire space with his liver. So the liver has the capacity for regeneration. It's part of the reason you can insult it so much, and it still lasts for you. That's it, right? Okay. Okay, so let's talk about skin aging. You know this is not going to be a good story, right? Okay. So what happens is your skin is working perfectly beautifully until you hit about your 20s. And then things start to slow down. Cell division starts to slow down. Protein regeneration of the collagen elastic fibers will start to slow down. So your cells will start to be less active. Dermatitis becomes more common. Dermatitis is just this irritation or inflammation of the skin, and it becomes more common because I have fewer Langerhans cells. What were those again? Immune system cells. I also have less oil, right, to protect myself. So you get dermatitis more often. You have less lubrication. The only good part about that is if you were a teenager who hated your oily skin in your 40s, you're going to look better than the rest of us because you get to keep oil for a little longer than the rest of us do, and it keeps your skin looking youthful and moist. Your collagen and your elastic fibers will degenerate and not replace as readily. So you break them down via gravity and UV radiation and everything else that you do to your skin. And until you're in your 20s, you replace them at about the same rate as you lose them. And then later, you just don't replace them as readily, which means that your skin is going to lose its elasticity and pliability. So if you pinch an 8-year-old, their skin snaps back immediately. If you do it to a 40-year-old, it might pause. And if you do it to an 80-year-old, it might still be up there tomorrow, right? It just like has hardly any elasticity and pliability. So in addition, if you lose collagen, this is going to make your skin thin, right? So the dermis is going to thin and the epidermis is going to thin. The epidermis thins because exfoliation happens at a relatively constant rate, but cell division slows, so your epidermis gets thin. And then the dermis thins because you're going to lose most of your connective tissue fibers sort of slowly and progressively. So in the very, very elderly, I don't know if any of you guys have worked at an old folks home, you have to be careful with how you move them because you can rip their skin, just rip holes in their skin if you're not careful with it. So your melanocyte production will decrease. What's that going to do? Your skin's going to get lighter. Your sweat production decreases, which means that just trouble thermoregulating. If an elderly person gets really hot, it's hard to cool down. Your vitamin D production will decrease, which affects everything that's dependent on calcium, right, including your bones. And then your body fat deposition will change again, right? Your body fat deposition will change again primarily due to your sex hormones changing. And your skin thins, and then the thing that now what we're going to talk about is I want to show you what the sun does to this whole story. So the sun and what we call photoaging. Dermatologists say that the 90% rule is in play, and that is that 90% of skin aging is due to sun exposure. So did I show you guys the truck driver picture yet? I showed? Okay. So this guy's a truck driver. Did you guys see this one already? No? Okay, so this guy's a truck driver. I bet you can figure out which side of his face the sun was hitting sort of constantly. This is an amazing study because you couldn't get a better control, right? Okay, except unless the other side was completely masked all the time, right? So because of course you have the same diet, you have the same genetics, right? You have all of the same environmental factors, and this side just got a tremendous amount more sun exposure. So what sun exposure does is breaks down your collagen and elastic fibers in addition to increasing your likelihood of skin cancer. That's a separate sort of thing to consider. So, and then in addition, there is a town, I can't remember what state it's in, it's called Twinsville. It's one of those I states. I'm terrible at geography. Idaho, Iowa, Illinois, I don't know, one of those places. Every year they have a national convention of twins, and so some brilliant scientists figured out this is a really great place to go and get some control studies and to show you what sun does in a pair of identical twins. You can see the difference here. So look at the dramatic increase in wrinkling. These are genetically the same genes, right? She had a lot of sun exposure and she was very limited in her sun exposure, and so you can see the dramatic wrinkling, you can see how hooded her eyes are compared to hers, and you can see the hyperpigmentation, and then look at them from a side view to see. So this is what the sun does to your skin. It breaks down your collagen elastin. It eventually causes hyperpigmentation as it starts to make your melanocytes behave differently. And then if you get really, really, really extreme about it, you can get to this point where you look like a freaking saddle. All right, and ain't nobody wants that, right? And that kid is like, what? That's how we're all feeling about that particular thing. But I lived in Florida for a while, and there's a lot of this going on. Go ahead.
My family and I, we go down to Lake Powell every year, and next to us is the same boat.
Over.
Same people, they live on the boat, and the lady looks exactly like that.
Yeah.
Exactly, if not worse, like just leather.
And still rocking that bikini, huh?
Still rocking. Still rocking.
Well, you know, more power to her until she gets skin cancer, right? Rock that bikini as long as you want to, honey. You are going to die, though. Okay, so, and then we aging in dark skin versus light skin people, you've all seen that, and I think we've talked about it. If you have inherently darker skin, then it protects your collagen and elastic fibers from UV radiation. And so you can see a person of African descent in her 60s, and she's still going to have a lot of the elasticity and collagen in her skin. And then if you have relatively fair skin and sun exposure, then you are going to start to break down. So that's your skin. The other thing that I want to tell you about aging is one other thing that we know dependably breaks down your collagen and elastic fibers besides UV radiation. What is it? Smoking, yeah. Smoking breaks down your collagen and elastic fibers incredibly. So if you both smoke and are a sun worshiper, you are going to look like a saddle in your 30s. So you better have all your hot stuff. Do it now. Just do it now, right? Because it's almost over. Okay, so briefly, let's talk about burns. When you hear about burns, you always hear about first, second, and third degree burns. So let's talk about that. What does that have to do with the layers of the skin that we've talked about in this set of notes? A first degree burn, by definition, only damages the epidermis. It can irritate the dermis, but it didn't damage the dermis, okay? And it doesn't have to be sun. It could be chemical. It could be steam. It could be any number of things. Now, the deal with sun is that if you damage with sun, you have both damaged the tissue and exposed it to UV radiation, right? So it's a double whammy. That's why that one's a big deal. So with a first degree burn, you only damage the epidermis and you could irritate the dermis. And generally it just manifests as redness in the skin, no blistering, okay? And so that is a first degree burn. And I've had them from like steamy pots and from the sun and from everything. And if you have a second degree burn, the second degree burn by definition damages the entire thickness of the epidermis and top of the dermis. And you're going to have blistering associated with the second degree burn. Okay? And so when you have blistering associated with a second degree sunburn, it's a big deal because we know we have damaged with UV radiation the entire epidermis and part of the dermis, which means you know that you've actually gotten damage to the collagen fibers, right, which will result in skin aging. But in addition, you also know you've gotten UV radiation to those bottom layers of the epidermis, which are the ones that have a high rate of mitosis, right? So you know you've gotten it all the way down there. And it's the blistering sunburns. I told you guys that if you have 2 or more blistering sunburns, then you increase your lifetime risk of skin cancer by around 250%, especially if you have them as a child. So interestingly, you know that regardless of how you get a blister, but let's say it's a blister from a burn, the top layer of the blister doesn't have any sensation, right? You could poke it with a pin. I'm not saying to do that, but you could poke it with a pin. And the reason is because when you get a blister, it is actually that inflammatory fluid is building up between the dermis and the epidermis. And when you do that, even if you had any of the tactile cells, right, they get pulled away from the nerve ending, right? So there's really no sensation in that upper level, right? And of course, most of it doesn't have any sensation anyway, but even if you had any, it gets pulled away from the nerve ending. But of course, immediately under that would be the top layer of the dermis, and that would hurt like crazy if you touched it, if you got under there. Does that make sense? So a second degree burn damages the epidermis and the upper dermis. And then a third degree or full thickness burn is one that damages the entire epidermis and the entire dermis. Hold on. It can even get deep enough that it crackles the fat, like cooking the fat. It's gross to think about, but it totally can happen. And scar tissue formation is very, very likely with third degree burns. Regeneration will happen at the edges, but you are at such high risk of infection that fibrosis or scar tissue formation is very likely to occur. I'll talk about how they sort of try to get around this in just a second. And with third degree burns, what you are going to have is the skin is going to be massively damaged. In the early stages, it looks like this. It can even look blackened, like the skin looks like it's actually been cooked. And third degree burns, people who have third degree burns, how do they estimate the amount of the body? They don't measure because they're already in a tremendous amount of pain, right? And you don't want to touch them in any way that's non-sterile. There's just an estimating mechanism based on an average size adult. And so this average size adult, each leg is about 9%, and the torso anterior is about 18%, posterior is about 18%. You have to use a totally different estimation mechanism for kids, right? Because kids have very different proportions, like their heads are enormous compared to the size of their little bodies. That's why they're so cute. So they estimate. Now let's talk about what they do. There's a whole bunch of research that goes into how to treat burn victims. And what you're trying to do is you're trying to keep them alive, of course, assuming that they have a large area of their body that's burned, and you have lots of infection risks and things like that. But in addition, of course, you want to have as much regeneration and as little fibrosis as possible, especially at joints, right? Because if you have fibrosis at a joint, it limits mobility at a joint. So historically, what they have done is let healing go on, and then if you start having fibrosis, they abrade it off, and then let the healing go back on again. And it is as horrible as it sounds, although I haven't experienced it. Did you experience that? Oh, I'm sorry. But now there are some ways that are less painful, even though they usually don't preclude the whole abrasion thing. So there are skin grafts and a bunch of different ways that you can do skin grafts. The first one is an autograft. That is a graft from a part of me, right, to a part of me, okay? And if the autografts are small enough and thin enough, then you don't get as much fibrosis in either place. You don't necessarily have fibrosis from where you took it, okay? It gets a little hard when you have children, right, because they don't have tremendous expanses of skin anyway. So autograft would be from me to me. And then an allograft, there's a bunch of different ways of doing allografts now. Historically speaking, they used to take the foreskin of little babies that were circumcised, little baby boys, and they didn't use the foreskin skin on burn victims, but they used those cells to grow up excess skin cells in the lab, right? They're nice and regenerative, they grow really well. So you use those cells and grow things up like big sheets, thin sheets of skin cells. But now, more commonly than doing that, they use cadaver skin. And cadaver skin, you take the cadaver skin, and what you do is you run it through, so you take a little square of cadaver skin and you run it through kind of like a combination of a steamroller and a meat slicer, so that you get really, really thin layers of cadaver skin. And then what they would do is, if I had a large wound, right, they'd put the cadaver skin in, but it's a placeholder. What it does is holds the place in the middle so that you can get regeneration at the edges, and then you remove it and you put a smaller piece in. And it holds the place so you can get regeneration at the edges, and then you remove it and then you put a smaller piece in. So it allows more time for regeneration without as much fibrosis occurring. Does that make sense? Okay. Now, do they end up with appreciable numbers of cadaver skin cells in them? No, not really, right? You can't say zero. But really it's just a placeholder that keeps fibrosis from occurring. And now they just came up with another technique, and I don't know if this is being done everywhere now, but they actually figured out a way to aerosolize skin cells. So they grow up skin cells in the lab and they put them in solution, and then you can actually spray them onto the surface. Isn't that freaking cool? Science! You spray them onto the surface, which decreases the likelihood of fibrosis and increases the likelihood of regeneration. Heterografts they don't do very often in this country. Heterografts are grafts from another organism, a pig, for instance. We don't do them very often in this country. They still do in other countries. And then there's also artificial skin, and artificial skin is usually a combination of artificial and real. So for instance, if I had a large wound, I could use an artificial matrix of connective tissue that's sort of like, you know how if you have a hole in the wall and it's a big hole, you have to put in the mesh and then you put the spackle over it. So what you've got is this mesh that you put in that will eventually disintegrate. Eventually the body will disintegrate the mesh, and then you just put skin cells over the top of it, and the healing process occurs more readily that way. So there's a bunch of advances in treating burns that are sort of consistently going on. Any questions? Everybody good? Okay, we're done for today. We're going to start nervous system next time. I redid your nervous system notes. The first set are done. I'm working on the second set. So I'm not going to give you guys a quiz next week because I'm going to let you concentrate on lab. However, if nobody comes on Tuesday, I'll give you a quiz on Thursday. If you guys decide to miss lecture,
I'll be here. I'll be here because